Hello and welcome. The deep tendon reflexes are one of the most clinically powerful parts of the neurological examination. It is a foundational bedside tool, one that will sharpen your diagnostic thinking from your neurology rotation forward in every neuromusculoskeletal specialty where careful clinical examination matters. My name is Dr. Conwell. What I want you to leave with is not a list of facts, but a mental framework you can rely on in the examination room. When you tap a tendon and something unexpected happens or fails to happen, you will know exactly what it means and what to do next. This is the first of a three-part series on deep tendon reflexes, a complete review of the neuroanatomy, the neurophysiology, and the neuropathology, finishing with the clinical examination itself. Module 1 builds the foundation, what a deep tendon reflex is, and the anatomy of the three reflex arcs. Module 2 adds the physiology, the spinal cord levels, the Wechsler grading scale, and interpretation. Module 3 brings it to the bedside, pathology, localization, and examination technique. For this first module, hold one goal in mind. By the end, you should understand what a deep tendon reflex is and be able to trace the circuits that produce it. Everything clinical we do later rests on this foundation. Let us begin. Here is the roadmap for Module 1. We open by defining the deep tendon reflex, its purpose, and the monosynaptic principle that makes it so fast and so reproducible. From there, we move to the anatomical reference, the neural components of the three reflex arcs, the myotatic stretch reflex, reciprocal inhibition, and the inverse, or autogenic reflex. We then walk the myotatic reflex arc step by step from the tendon tap to the muscle contraction you observe at the bedside, and we close the module with the companion reflexes that work alongside the stretch reflex to keep movement smooth and protected. We begin with Chapter 1, the foundation. The first question we have to answer is the most basic one. What exactly is a deep tendon reflex, and why does the nervous system bother with it at all? I want you to hold that question in mind for a moment before I give you the answer, because your understanding of the DTR will be much richer if you appreciate the physiological problem it solves. The nervous system is faced with a continuous challenge. How do you maintain upright posture and controlled movement in a body where muscles are constantly changing length, constantly dealing with unexpected loads, and constantly at risk of being overstretched? The deep tendon reflex is one of the nervous system's most elegant solutions to that problem. Let's look at the definition and the mechanism. Here is the formal definition, and I want you to pay attention to every word. A deep tendon reflex is a monosynaptic spinal reflex, specifically a muscle contraction produced in response to a sudden stretch stimulus. That's the myotatic stretch reflex. Now the word monosynaptic is doing important work here. Mono means one. A single synapse separates sensory input from motor output. One synapse, that's it. The signal enters the spinal cord on the sensory side, crosses one synapse, and immediately activates a motor neuron. This is the simplest possible wired circuit in the nervous system, and that simplicity is what makes it both fast and diagnostically reliable. What is the purpose? Three things. First, it maintains posture and steady muscle tone, a continuous background function you're not consciously aware of. Second, it protects the muscle from injury by resisting sudden overextension. And third, it operates as an automatic length control system. The muscle is essentially monitoring its own length in real time and self-correcting. How do we elicit it clinically? A brief tap on the tendon with a percussion hammer. That tap briefly stretches the muscle, which activates the stretch receptors, which triggers the reflex arc. And here's the clinical bottom line. Because the circuit is short and predictable, the DTR gives us a fast, reproducible window into the integrity of a specific spinal segment and its peripheral nerve. That's why we test it at the bedside. In one line, stretch the muscle and it contracts back, a built-in protective reflex that doubles as a diagnostic tool. Take a moment to study this diagram carefully. What you're looking at is the complete myotatic stretch reflex arc. from the initial stimulus all the way to the observable muscle contraction. I want you to trace the pathway with your eyes as I walk you through it. The stimulus begins at the tendon. The tap stretches the muscle, which activates the muscle spindle, the stretch receptor embedded within the muscle belly. From the spindle, a signal travels along the 1A afferent fiber, a large, heavily myelinated sensory nerve, into the spinal cord via the dorsal root. Inside the cord, the 1A afferent makes a direct excitatory synapse with the alpha motor neuron. That's the one synapse that makes this monosynaptic. Simultaneously, it activates an inhibitory interneuron, which we'll return to when we discuss reciprocal inhibition. The alpha motor neuron then sends a signal back out through the ventral root to the muscle, which contracts, and that contraction is the reflex response you observe clinically. This is the anatomy of what happens every time you tap a tendon. Commit this pathway to memory. Everything else in this lecture is built on top of it. Chapter 2 takes us into the anatomy in greater detail. I want to introduce you to 3 reflex arcs that function together in the nervous system, and then we'll examine the neural components of each one carefully. The first is the deep tendon or myotatic stretch reflex, the primary subject of today's lecture and the one you test with a reflex hammer. The second is the reciprocal inhibition reflex, a companion reflex that runs simultaneously with the myotatic reflex and ensures that when one muscle contracts, its antagonist relaxes. Without this, movement would be rigid and uncoordinated. The third is the inverse-autogenic myotatic reflex, also called autogenic inhibition, a protective mechanism mediated by a different receptor, the Golgi tendon organ, that prevents muscle injury when tension becomes excessive. These 3 reflexes work together as an integrated system. You need to understand all 3 to fully interpret what you're seeing at the bedside. Let's look at each one in turn. This slide presents the anatomical components of the myotatic stretch reflex arc, and I want you to be able to name and describe each component from memory before we leave this section. The key players are the muscle spindle, the receptor that detects stretch, the IA afferent fiber, the large myelinated sensory neuron that carries the signal to the cord at speeds of 80 to 120 meters per second, the dorsal root where the sensory signal enters the cord, the single synapse in the ventral horn of the spinal cord gray matter, the alpha motor neuron, the motor neuron that fires in response, and the ventral root through which the motor signal exits the cord and travels back to the muscle. Study the anatomical arrangement here. Notice where each component sits. The sensory side on the dorsal aspect, the motor side on the ventral aspect. This dorsal sensory ventral motor organization is one of the most fundamental principles in neuroanatomy, and it applies throughout the spinal cord. Now we add the second reflex arc, reciprocal inhibition. Notice how this diagram builds on the previous one. The IA afferent still enters the dorsal horn. It still makes the direct excitatory synapse with the alpha motor neuron of the agonist muscle. That part is unchanged. What's been added is this. The same IA afferent also connects to an inhibitory interneuron. That interneuron reaches across to the alpha motor neuron of the antagonist muscle and inhibits it, simultaneously with the agonist being activated. Agonist contracts, antagonist relaxes at the same time from the same sensory signal. This is the elegance of the nervous system's design. You don't need 2 separate commands. One tendon tap activates contraction in one muscle and simultaneously suppresses the opposing muscle. The result is smooth, coordinated movement. Keep this in mind for later. When we discuss upper motor neuron lesions, the loss of reciprocal inhibition contributes directly to spasticity. The third arc, autogenic inhibition, also called the inverse myotatic reflex. This one involves a different receptor entirely, the Golgi tendon organ, or GTO. Where the muscle spindle monitors muscle length, the GTO monitors muscle tension. Specifically, it detects how hard the muscle is pulling. When tension rises to a dangerous level, the GTO fires. Its signal travels along the IB afferent fiber. Notice on this diagram that the IB does not make a direct synapse with the alpha motor neuron. It requires an inhibitory interneuron. That makes it polysynaptic in contrast to the monosynaptic stretch reflex. The result, the alpha motor neuron of that same muscle is inhibited and the muscle relaxes. The tension is released before the tendon can be avulsed. This is why it's called autogenic. The muscle inhibits itself, and why it's called inverse. The myotatic reflex causes contraction, while this reflex causes the opposite, relaxation. This mechanism is also clinically relevant in UMN pathology, where its disruption contributes to a phenomenon called the clasp knife response, which we'll come back to. Chapter 3 takes the anatomy we've just reviewed and walks us through the complete myotatic reflex arc and sequence, step by step, from stimulus to response. This is the core mechanism. Everything in clinical reflex testing is an application of this pathway. I want you to be able to recite these steps in order, understand what's happening at each one, and know what a disruption at any point would look like clinically. Let's work through it. 7 steps. Follow along. Step 1, tendon tap producing muscle stretch. The percussion hammer strikes the tendon, creating a brief, sharp stretch of the muscle. This stretch is the physical stimulus that initiates the entire reflex sequence. Step 2, muscle spindle activation. Inside the muscle belly, the muscle spindle, the length sensing receptor, detects this sudden elongation. It responds by generating an electrical signal proportional to the rate and magnitude of the stretch. Step 3, the IA afferent fiber. That signal is immediately carried by the IA afferent, one of the largest, most heavily myelinated nerve fibers in the body, toward the spinal cord. Because of its heavy myelination, conduction is fast, 80 to 120 meters per second. Step 4, the spinal cord synapse. The IA afferent enters the cord through the dorsal root and makes direct excitatory contact with the alpha motor neuron in the ventral horn. Simultaneously, and I want to emphasize that word simultaneously, it activates an inhibitory interneuron. Step 5, interneuron function. That interneuron reaches across to the antagonist muscle's alpha motor neuron and inhibits it. This is reciprocal inhibition happening in real time, concurrent with agonist excitation. Step 6, alpha motor neuron response. The alpha motor neuron fires, sending an efferent signal back down through the ventral root into the peripheral nerve to the muscle. Step 7, muscle contraction, the observable DTR response. The muscle contracts briskly, producing the tendon jerk you see at the bedside. That is the full circuit. 1 tap, 7 steps, a fraction of a second. Let's focus specifically on the muscle spindle because it is the critical receptor at the heart of the DTR. The muscle spindle is an encapsulated stretch receptor embedded within the muscle belly, running in parallel with the surrounding extrafusal, that is regular, muscle fibers. Inside the spindle capsule are modified muscle fibers called intrafusal fibers. These intrafusal fibers don't contribute meaningfully to muscle force, but they are exquisitely sensitive to changes in length. There are 2 types of intrafusal fibers you need to know: nuclear bag fibers, which detect the velocity of stretch, and nuclear chain fibers, which respond to sustained stretch. Both are innervated by 1A afferents at their central equatorial region. Critically, the ends of the intrafusal fibers are also innervated by gamma motor neurons. We'll come back to those shortly, but keep in mind that the spindle's sensitivity can be actively tuned by the nervous system. It is not a passive sensor. Look at the projections on this diagram. The IA afferent exits the spindle's equatorial region and heads directly to the spinal cord. This single fiber, fast, myelinated, direct, is the express lane of the reflex arc. Now let's look at the other critical element, the alpha motor neuron. The alpha motor neuron is the final common pathway for all voluntary and reflex motor commands. Every signal, whether it originates in the motor cortex, the cerebellum, the brainstem, or the spinal cord itself, ultimately must pass through the alpha motor neuron to reach the muscle. That is why it is also called the lower motor neuron, and why damage to it produces such characteristic clinical findings. Its cell body sits in the ventral horn of the spinal cord gray matter. Its axon exits through the ventral root, travels in a peripheral nerve, and terminates at the neuromuscular junction, where it releases acetylcholine to trigger muscle contraction. One alpha motor neuron, together with all the muscle fibers it innervates, forms a motor unit, the smallest functional element of movement. Remember this: when the alpha motor neuron or its axon is damaged, you lose the output of the reflex arc. The signal can't complete. That's why LMN lesions produce absent or diminished reflexes. Now we come to one of the most clinically important concepts in this lecture: the mapping of individual reflexes to specific spinal segments. Here is the core principle. Each deep tendon reflex is mediated by a specific set of nerve roots. The muscle spindle's 1A afferent enters the cord at a known spinal level. The alpha motor neuron that fires in response exits at the same level. This is why a reflex can be used to localize a lesion with precision. The major reflexes you must know cold, and I mean reflexively, are the biceps, the triceps, the brachioradialis, the patellar, and the Achilles. Each one maps to specific cervical, lumbar, or sacral segments. Here is the immediate clinical application. If a patient has a diminished patellar reflex, that points to L3 to L4. Diminished Achilles, S1. Diminished biceps, C5 to C6. The reflex itself is telling you the address of the problem. The detailed root level tables follow on the next slides, and those tables are worth your time. But the principle to internalize now is this. Reflex loss localizes the lesion. Reflex exaggeration tells you the lesion is above the level of that arc. We'll build on both of these shortly. This is your pictorial reference for the reflex arc components we've covered so far. Take a moment here. Rather than moving quickly to the next concept, I want you to use these images as a mental consolidation point. Look at each structure, name it in your mind, and connect it to the function we just discussed. Muscle spindle, length sensor, generates the IA signal. IA afferent, fast, heavily myelinated monosynaptic connection to the alpha motor neuron. Alpha motor neuron, lower motor neuron, final common pathway, cell body in the ventral horn. The synapse. One synapse in the ventral horn between 1A, afferent, and alpha motor neuron. The efferent, alpha motor neuron axon traveling to the muscle via ventral root and peripheral nerve. If you can picture all of these in anatomical relationship to one another, you're ready for the next section. Chapter 4 introduces the two companion reflexes that work alongside the myotatic stretch reflex. We've already touched on both of these in the context of the anatomy slides, but now I want to go deeper into the physiology of each one, how the circuit works, what it accomplishes, and what happens when it's disrupted. The two reflexes are reciprocal inhibition and the inverse-autogenic myotatic reflex. Together with the myotatic stretch reflex, these three form an integrated system that gives the nervous system fine control over movement, posture, and protection from injury. Let's take them in turn. Reciprocal inhibition. Let's trace both limbs of this reflex together. The excitatory limb first. Step 1. A tendon tap or voluntary movement stretches the agonist muscle and activates the muscle spindle. Step 2. The IA afferent, large myelinated, carries the impulse to the spinal cord via the dorsal root. Step 3. The IA makes a direct excitatory synapse with the alpha motor neuron of the agonist. The agonist contracts. Now the inhibitory limb, and this happens simultaneously. Step 4. The same IA afferent also synapses with an inhibitory interneuron in the spinal cord at the same time it's exciting the agonist motor neuron. Step 5. That interneuron inhibits the alpha motor neuron supplying the antagonist muscle. The antagonist relaxes. The net result? Agonist contracts, antagonist relaxes in a perfectly coordinated, simultaneous response to a single sensory input. Why does this matter? Without antagonist inhibition, every contraction would meet resistance from the opposing muscle. Movement would be rigid, jerky, and metabolically exhausting. With reciprocal inhibition, the nervous system ensures that as a muscle contracts, its opposing muscle gets out of the way. And here is the clinical pearl. Loss of reciprocal inhibition, which occurs in upper motor neuron lesions, is a significant contributor to spasticity. When the inhibitory interneuron circuit is disrupted, the antagonist doesn't yield properly, and you get that characteristic increased resistance to passive movement. Look carefully at the color coding on this projection diagram. Blue for the 1A afferent sensory pathway, red for the excitatory motor pathway to the agonist, and green for the inhibitory interneuron pathway to the antagonist. Follow the blue 1A afferent as it enters the cord and branches. One branch, shown in red, goes directly to the alpha motor neuron of the agonist. The other branch, shown in green, goes to an inhibitory interneuron, which then reaches the antagonist motor neuron. The spatial arrangement matters. The excitatory and inhibitory connections happen within the same spinal segment. This is a local circuit, operating at the level of the cord, not requiring input from the brain. That last point is important. The brain doesn't initiate reciprocal inhibition. It happens automatically as an intrinsic property of the spinal cord circuit. The brain can modulate it, and does, but the basic circuitry is hardwired into the cord itself. Now, the inverse myotatic reflex, autogenic inhibition. This reflex depends on a completely different receptor, the Golgi tendon organ, or GTO. The GTO is located at the musculo-tendinous junction, arranged in series with the muscle fibers, meaning it senses tension rather than length. It doesn't care how long the muscle is, it cares how hard it's pulling. The sensor here is the IB afferent fiber, also large and myelinated, but distinct from the IA. Here is the critical anatomical distinction. Unlike the IA, which synapses directly on the alpha motor neuron, making it monosynaptic, the IB must route through an inhibitory interneuron that makes this reflex polysynaptic. The circuit, excessive force activates the GTO, which fires the IB afferent, which activates an inhibitory interneuron, which inhibits the alpha motor neuron of the same muscle, which causes the muscle to relax and the tension to drop. This is the autogenic part. The muscle is inhibiting itself, and it is called inverse because where the myotatic reflex causes contraction, this one causes relaxation. The clinical significance is directly practical. This is a protective mechanism that prevents tendon avulsion. The GTO is a biological safety brake, and when this mechanism is disrupted in UMN lesions, specifically when the IB inhibitory circuit is dysregulated, you get the clasp knife phenomenon, an initial high resistance to passive stretch that suddenly gives way like a closing penknife. That sudden yield is a loss of this protective relaxation mechanism. Here is the anatomy of the Golgi tendon organ in detail, located at the musculo-tendinous junction. The GTO is a capsule of collagen fibers interdigitated with the terminal portion of the muscle fibers as they transition into tendon. The 1B afferent nerve fibers are braided through this collagen matrix. When the muscle contracts and pulls on the tendon, the collagen fibers of the GTO are mechanically deformed, which activates the 1B afferent endings embedded within them. This is a tension sensor, not a length sensor. Compare this to the muscle spindle, which sits within the muscle belly, running in parallel with the muscle fibers. The spindle is loaded when the muscle lengthens. The GTO is loaded when the muscle contracts. These two receptors are therefore complementary. Together, they give the nervous system both length information and tension information in real time. Note that the GTO has a much higher threshold than the muscle spindle under passive stretch conditions. It requires active contraction to fire reliably. This is why the IB-mediated reflex doesn't interfere with every small movement. It only engages when tension reaches a level that risks tissue injury. This diagram shows the full IB pathway for autogenic inhibition. Trace it from the GTO at the musculotendinous junction, through the IB afferent into the dorsal root, and then into the spinal cord where the key feature appears, the mandatory inhibitory interneuron. The IB does not make a direct synapse with the alpha motor neuron. It cannot. It must route through this interneuron. That interneuron inhibits the alpha motor neuron of the same muscle, autogenic inhibition, and in some cases has additional projections to antagonist motor neurons that produce a corresponding excitatory effect on the opposing muscle. So when a muscle is forced to relax under excessive tension, its antagonist may receive a mild facilitatory signal. This makes physiological sense. If the biceps is inhibited to protect the tendon, a gentle signal to the triceps helps restore balance. Compare this projection diagram to the one for reciprocal inhibition. Notice the similarities in circuit architecture. Both use interneurons, both have simultaneous effects on agonist and antagonist, but the receptor, the afferent fiber type, and the triggering condition are all different. This is a comparison slide you need to commit to memory because the distinction between IA and IB is tested regularly and is clinically meaningful. Group 1A afferent fibers arising from the muscle spindle primary endings. The muscle spindle is a length detector. The receptor is the muscle spindle embedded within the muscle belly, running parallel to the extrafusal fibers. It detects changes in muscle length. Specifically, it fires when the muscle is stretched or elongated. The reflex it mediates is the deep tendon reflex, the myotatic stretch reflex. The pathway is direct. Stretch, 1A fires, 1 synapse, alpha motor neuron contracts the muscle. Clinical pearl. This is what you test with a reflex hammer. The tap stretches the tendon, which stretches the spindle, which fires the 1A. Group 1B afferent fibers arising from the Golgi tendon organ. The Golgi tendon organ is a tension detector. The receptor is the GTO at the musculotendinous junction. It detects muscle tension or force, not length. It fires most strongly during active contraction when load is high. The reflex it mediates is the inverse stretch reflex, autogenic inhibition. The pathway requires an interneuron. High force, GTO fires, IB afferent, inhibitory interneuron, alpha motor neuron inhibited, muscle relaxes. Clinical pearl, the protective safety brake. Loss of IB inhibition contributes to the clasp knife phenomenon in UMN lesions. The memory aid, 1A equals length, muscle spindle, 1B equals tension, Golgi tendon organ. That association will serve you on every board exam and at every bedside. Let's consolidate the comparison with this side-by-side summary. On your left, the muscle spindle. On your right, the Golgi tendon organ. Study the anatomical positions first. The spindle is within the muscle belly, running parallel to the extrafusal fibers. The GTO is at the junction, where muscle becomes tendon, arranged in series. In parallel versus in series is not just anatomy jargon. It tells you how each receptor is loaded. Because the spindle is in parallel, it gets stretched when the whole muscle is passively elongated. This makes it ideal for detecting length change. Because the GTO is in series, it gets loaded when the muscle pulls on the tendon during active contraction. This makes it ideal for detecting force. 2 receptors, 2 afferents, 2 reflexes, 2 functions, working together to give the nervous system a real-time picture of both what the muscle is doing and how hard it's working. This is one of those elegant examples of biological engineering. 2 sensors covering complementary aspects of muscle state, each triggering a protective reflex appropriate to its input. If this lecture reinforced the value of a rigorous, systematic approach to the neurological reflex examination, I want to make you aware of a resource that takes that approach well beyond what we've covered today. The Clinical Picture, Essentials of the Neuromusculoskeletal Exam, authored by Dr. Timothy Conwell, is a comprehensive clinical reference that covers the complete neuromusculoskeletal workup, the history and physical examination, neurological and orthopedic evaluation, clinical case studies, and documentation that includes narrative reports and SOAP charting. This is the only text that takes you from examination findings all the way through to a finished clinical record in a single, concise volume. It is available now on Amazon. The link is on your screen or below in the description. Amazon.com/DP/1540795-225. If you are preparing for board examinations, entering your clinical rotations, or simply want a reference that translates neuroscience into practical clinical documentation, this textbook belongs on your desk. Before you move on, I encourage you to reinforce what you've learned today by working through this module's complimentary board preparation flashcard set. It includes 50 board-style examination questions, each accompanied by a detailed explanation and rationale, and is available now at www.theclinicalpicture.com/lecture-series. Don't simply read through the answers. Challenge yourself to reason through each question before revealing the explanation. This process of active recall is one of the most effective ways to strengthen retention, sharpen clinical reasoning, and build the confidence you'll need for both your board examinations and the patients you will care for throughout your career. If you found this lecture helpful in deepening your understanding of this important clinical topic, please consider subscribing, liking, or leaving a comment below. This will greatly help me produce more content like this. Thank you for watching.